It's a pleasure for me to be here with you this morning to tell you a bit more about Orbit Biomedical. So we're a medtech company. We're not a gene therapy development company, but we're focused on a very important problem, which is to revolutionize the delivery of gene and cell therapy to the back of the eye. We were set up earlier in 2018, so we're very nearly a year old. And we were launched with Series A funding from Syncona, which is a UK-based life science investor based in Philadelphia. And we are unique in that we operate at the intersection of cell and gene therapy, surgical device technology, and surgeon training. And these are things that actually rarely intersect, but for delivering gene and cell therapy to the back of the eye, this is extremely important. So the company has been founded based on technology that was developed by Janssen in the context of a cell therapy program. And we're extremely fortunate to count amongst our founders the device technology experts who developed the technology that I will show you, but also a team of trainers who are expert at training surgeons all over the world and have plenty of expertise in doing this in other areas like hips and eyes and ears and other areas. So retinal diseases affect more than 20 million people in America. They can affect children, young ages, and also early adults. These are primarily inherited retinal diseases, but they also affect older adults in broader indications like age-related macular degeneration, diabetic retinopathy, which are huge problems. So the retina is the tissue that's at the back of the eye. It's a very fragile tissue layer, and it contains the cells that detect light signals and turn them into neural signals to send to the brain. So these indications where the retinal tissue is degenerated are associated with vision loss and in some cases even complete loss of sight, and they're extremely debilitating to patients. So there are a huge number of people that are affected. So age-related macular degeneration alone, for example, affects 11 million Americans. So this is a big problem. The good news is over the last few years, there have been some really exciting advancements in the field of gene and cell therapy, and there are a number of programs now that are in clinical development that offer promise for these patients, and there is clinical data now to show that these programs can slow down disease progression and in some cases even restore vision, so they're hugely impactful. And in fact, the first AAV gene therapy program to be approved here in the US is a program for a rare inherited retinal condition. So there's a lot of hope for these patients. However, there is a problem that still needs to be solved as we scale these programs from rare monogenic disorders and into much bigger indications. And that problem is the problem of delivery. So right now, gene and cell therapy delivered to the back of the eye is delivered into what's called the subretinal space. And it's delivered using a short surgical procedure called transvitreal subretinal injection. And what you can see here on the left is just a little diagram here that describes the procedure. So it's a 3-step procedure, and each one of these steps is well known to surgeons. They're trained on it and they use it routinely for other purposes, and they're very familiar with each of these steps. However, having been trained on it for other purposes is not the same as being able to deliver a small quantity of a very high-value therapeutic. And this procedure is not fit for purpose. It's not designed for purpose for this. And so, as I'll illustrate, it's associated with various issues which just make it challenging to deliver these kind of therapeutics. So the 3 steps are to remove the vitreous, which is the gel-like substance in the middle of the eye, to then use a needle to try and hook under the end of this very fragile tissue-like layer, which is the retina, which is shown in yellow here on the slide, and then inject the therapeutic into a bleb, which is the bubble that you can see there, and obviously withdraw the needle. And as you can see, it's not a stable process. You're trying to kind of hook a very fragile tissue on a pivot point, which is a way away from where you need to be. And withdrawing the needle is associated with egress of the therapeutic. So when you think about the implications of this, the drug that leaves the syringe is not the same as the quantity of drug that actually is reaching the subretinal space. So there's a great deal of uncertainty here about the dose. What dose is it that each patient is actually receiving in the subretinal space? There is also a lot of variability from patient to patient and surgeon to surgeon, and there are also various procedure-related safety effects that are important to consider here. So the challenge is, how do you take this, which is it's a well-established procedure, it's well-known, but how do you take it and scale it? Because when you're doing this in a small-scale study for an inherited retinal disease, that's one challenge. But when you want to extend this technology and make it applicable for those large indications like age-related macular degeneration, how do you scale this as a process? And that's what we have been focusing on. So the technology that we've delivered addresses the problem from a different way. So the team, and this was work that was done at Janssen, the team at Janssen sat down and considered the problem from first principles. And the solution that they've come up with is to use a flexible microcannula, which you can see here on the right, and it snakes in between the tissue layers, so it's very stable. It goes around the back of the eye, and it accesses the subretinal space from that position. And it has very clever technology in there, so there is a small microneedle, which you can see here. There's a lot of engineering around that microneedle, which enables the therapeutic to be delivered in exactly the right place. And what's important here is that we can do this without needing to make a hole in the retina. So you can see on the left-hand side, when you're trying to hook that tissue with the needle, you're making a hole in the retinal tissue, and often it gets pulled and it gets teared away. We can do it without making a hole in the retina. We can do it without removing the vitreous. And these things are important to the safety of the procedure. But what's more important is we can also deliver without losing the product into the vitreous, and this has all sorts of implications for the development of these therapeutics. So we're very happy that we have something. It's stable, it's precise, and it's simple, and it's been developed with input from many retinal surgeons to make sure that it's usable within the OR. So here on the right, you can see our commercial device. This is currently in development. Just some points to note. It's small. It uses the same technology to reach the back of the eye that I was just showing you, but it has been really optimized to make it highly ergonomic, easy for surgeons to use, and very safe for the patients. And we're testing this now with surgeons, and we've had a really fantastic response. They find this really exciting technology to use. So this is what we're working on currently. The value of this technology, though, goes beyond the device. So for us, and back to the slide that I presented at the beginning, the fact that we intersect across these three areas of understanding gene and cell therapy, understanding surgical delivery, and understanding surgeon training, these things are really important. So what goes hand in hand with the device technology is the knowledge to be able to train surgeons and to do this on a large scale so that we can result in consistent outcomes, whether it be through clinical trials or whether it be at commercial scale. And as soon as you can eliminate the inconsistency and the variability of delivery, then there are a whole bunch of benefits that flow from that. So if you can optimize the consistency of your clinical trials, then you can facilitate a faster and more effective development pathway. Because of the safety of the procedure, we can potentially access broader patient populations, and we've really designed this to be scalable. So what we want here is to take that surgical procedure and make it something that people all over the world can use for delivering these kinds of therapeutics. Another point which relates to that is if you can try and eliminate as much as possible of the translation risk from what you see in clinical trials to how your product is going to behave on a commercial scale and post-launch, that's also very important, especially in today's world of potential annuity models and or pay-for-performance reimbursement systems. So these things are all very important. We feel like we have a high-value technology, Our model is to work with companies that are developing cell and gene therapies, so we don't develop our own therapeutics ourselves, but we work with those companies to optimize the delivery of their products and optimize the clinical outcome of their programs. And our goal really is, as it says here on the photo with the devices, we want to try and enable this hugely transformative technology in gene and cell therapy, which has the potential to halt disease progression and potentially restore sight in many of these indications. We want to enable that technology to be as broadly as applicable to as many patients as possible, and that's really our goal. So we're early days, we're 12 months in, we have a lot of development work to do, but we're really excited about the potential, and we look forward to keeping you updated as we move forwards.
Thanks, Susan. That's really fascinating technology. So avoiding a vitrectomy really sounds like an improvement. Could you talk a little bit more about the side effects or the sequelae of doing a vitrectomy in patients who get SRIs?
Sure. Is this on? Yes. So a vitrectomy involves removing the gel-like substance that fills the eye. To call it a gel-like substance is really a bit of a simplification, because it's actually fibrous and it's connected to the retina. It's also translucent, so it's very difficult for the surgeons to see. So the surgeons, they look through the lens into the center of the eye. Normally it's lit up so that they can see what they're doing. They can't see the vitreous. And they quite often need to inject steroid, because the little steroid particles stick to the fibers within the vitreous, and then they can see where they are. So when they're removing it, they use essentially a vacuum to take out this substance. And they have to be extremely careful, because if they catch these fibers and pull on the retina and they pull the retinal tissue away, it's a little bit like taking wallpaper off a wall. If they pull the retinal tissue away, then they're separating the tissue layers, and it can result in vision disturbances and even loss of sight, and it requires additional surgery in order to connect it. So it's a routine procedure, but it's not without its challenges. It's tricky to do. There is also a lot of data in the literature which shows that removal of the vitreous is associated with a risk of cataract formation. So cataract is something which is really associated with older people. So you rarely see people younger than their 40s with cataract. But it is associated with this procedure. So when you think about the inherited retinal diseases that are being treated using this surgical approach, there is a risk that we will be creating a new population of young children, early teens, who form cataracts because of the surgical procedure. So this is something that it would be good to avoid, and there is a way to avoid it. The other point about the vitreous is it changes over time. So the vitreous in children is much more closely attached to the retina. So for young children with inherited retinal diseases, if you want to treat them below the age of 10, it's actually technically very challenging to remove the vitreous because it is so closely attached to the retina, and many adult retinal surgeons, are not happy to do that procedure. They want to refer to specialists, and even they are rather concerned about doing it. So it's got its challenges, and being able to remove that step from the procedure, I think, results in some quite substantive benefits.
Right, so that's safety. On efficacy, your presentation mentioned the efflux of therapeutic with traditional subretinal delivery, and that leads to variability. Has that been quantified? And so how much efflux is there, how much variability, and perhaps what do you hope to achieve with your device?
Yeah, that's a great question. So we know that there is efflux, and we know through a whole variety of mechanisms. So first of all, if we put a fluorescent solution and deliver it using the current technique, we can see the efflux. It's obvious, and it's caused by a variety of things. It's not one needle hole that makes that subretinal bleb. In reality, there's a lot of movement. There are normally multiple holes in the retina, and there's pressure that's forcing the fluid to flow back out. So we can see it with fluorescence. We know that it's happening in clinical trials because, for example, in some of the cell therapy studies, when cells leak out of the subretinal space and are present in the vitreous, they cause what's called epiretinal membranes, which is like a fibrous membrane that forms across the retina, and it's associated with vision disturbances. It requires another surgery to remove it. So we know that that's caused by cells that are leaking out into the vitreous. We also know because, we talk to the retinal surgeons that are running many of the studies involving subretinal injections, and they tell us that it's a problem for them. In terms of quantifying it, there was a great study that came out last year from Duke, from Cynthia Toth's group, and they have OCT imaging technology. They've developed a way of being able to very accurately measure the volume of the bleb. So the surgeon, when they look down the microscope, they can see the shape of it, but they can't very accurately estimate the volume of solution that's in there. And so they have a way of being able to do that. And they ran a small study in a porcine model where they measured the volume of the bleb in the subretinal space versus a target volume that was to be delivered. So of a target volume of 50 microliters that they were delivering, they found on average only 20 microliters of it was in the subretinal space. So that translates to around 60% of the volume that they were trying to deliver was not reaching the subretinal space. So, we don't have it quantified precisely in the clinical situation yet, but, there are many reasons to believe that there's quite a substantial loss of product, and that's obviously something that we're keen to investigate further.
Great. I think we're about out of time, but just very quickly, as I understand, the technology came from Johnson & Johnson's Janssen and had been tested in 21 patients or so. Please correct the number if that's wrong, but what advantages does that sort of give you in terms of picking up a technology that had some investment in it?
Yeah, so we're very fortunate to be able to build on that. So Janssen took the device into a clinical trial in 21 dry AMD subjects. And they ran the study across 8 different surgical centers, and they used 8 different surgeons, not because they needed to do that to recruit the subjects, but more because they wanted to have 8 surgeons involved in the training and in using the device. And the study was a usability study, so it was looking at using the device, and it was also looking at the safety of the cell therapy product that was being delivered. And it generated some great data. So through that clinical data, we know that we can use the device to deliver successfully to the subretinal space. We know that it's possible to do it without removing the vitreous. We know that it doesn't pierce the retina. We also know that in that study there was no egress of cells out into the vitreous. And we also know from ongoing follow-up from the study that there's been no incidence of cataract from that study as well. So we have a really good foundation in terms of the safety data, and there were also learnings around the device and the training that came out of that too. So for us, the challenge now is to take that forwards into our final commercial device and to really focus on how we can optimize the performance of the product beyond the safety of the procedure itself. So we're lucky to have access to that.
Great. Thank you, Susan, for the very clear presentation.
Thank you.